Hi, we are Burn Victim Bioink. My name is Michael, and we also have Kelsey and Maddie. For just a quick background, in the United States, around 14,500 people die every year due to burn-related injuries. These are usually caused by second or third degree burns. Now, the current treatment for these are usually skin grafts. But with our research, we've thought that we could possibly give another alternative to the source of these skin grafts. And that's why this is important, is because burn victims need a biocompatible, cheap, and personalized skin graft source to allow for optimal healing. Now, the goals for our project are: 1, to design a procedure to create a decellularized extracellular matrix, or DECM for short, out of murine epithelial tissue. And our second goal was to design a procedure to create a bioink using DECM to bioprint skin for burn victims. Now, the reason why we use decellularization was because it's very biocompatible. It removes cellular components and preserves the ECM and microstructure of the tissue.
In combination with decellularization, bioprinting is a valuable technique. Bioprinting is useful because it allows the 3D printing of tissues and organs using hydrogel bioinks. Bioinks are advantageous in tissue engineering because the printed DECM is an optimal microenvironment to support tissue organization. Based on the background information and goals of our project, we created this diagram to show the steps within the decellularization and bioprinting processes to end with an implantable skin graft. The first process of decellularization creates a DECM. On the left is a native skin tissue, and on the right is a decellularized skin. We designed a procedure using trypsin, dispace, and SDS to break open the ECM and wash away the cells and DNA. This leaves behind the structure of skin made up of keratin, fibrinogen, collagen, and elastin. To find the optimal procedure, we performed 3 trials testing the length of time, percent of SDS, and agitation period. We analyzed these results using the electron scanning microscope to look at surface, roughness, and topography. This diagram shows the 3 trials at 1, 5, and 10% SDS at a magnification of 200. Based on these results, we determined the 1% SDS with a 48-hour soaking time with agitation has the most ideal roughness without showing destruction of the ECM. Ideally, if we had more time, we would have performed completed histology tests and DNA quantification tests. The histology test would show the number of cells left in the decellularized skin. The DNA quantification test would analyze the percent of DNA remaining in the samples. We would expect less cells and DNA and the higher concentration of SDS and longer soaking and agitation periods. The other procedure we needed to create was for the bioink. It was important to consider what is expected for an ideal bioink in order to optimize 3D structure. It is important to consider layer height, extrusion flow rate, printing temperature, and speed. For the additives for the ink, we needed to think about regenerative abilities, viscoelasticity, cell biocompatibility, and biodegradation. We researched many mixtures to create an ink suitable for our needs and found that a blend of agarose, collagen, and fibrinogen would be best. With our research on these additives, we began creating an efficient gel preparation protocol. We had trouble refining the DECM powder enough for mixing, but ideally our DECM would have been a very fine powder. It was important to realize how properties may change with the addition of the powdered DECM, which is why ideally we would have tested flow rate before the addition of the powder and after. Due to the COVID-19 lockdown, we were unable to finish testing, so we based our expected results off of literature research. One text that was particularly helpful was titled "Printability Study of Bioprinted Tubular Structures Using Liquid Hydrogel Precursors in a Support Bath." They found that the ideal temperature when printing gelatin alginate hydrogel is 37 degrees Celsius, and this was because there was no clogging in the extrusion needle, there was steady and constant flow rate, and the temperature was compatible for cell viability. We used a lot of their research to help guide us to predict our expectations. Since their tests were done in a support bath, but ours would be done in an air medium, we would expect the filament cross-sectional area to increase as the flow rate increases and the travel speed decreases. In result, we would expect to perfect the procedures, and gather more results if we did not encounter the COVID-19 shutdowns. Any questions, please email us.